Welcome once again to Population Genetics and Evolution Bio 208 Unit 3, Session 6, How Evolutionary Principles Improve the Understanding of Human Health and Disease. And by the end of this session, you should be able to outline the basic evolutionary principles towards human health and disease and discuss how evolutionary principles could improve human health and disease management. So, basic principles of evolutionary medicine. While medical practitioners and public health specialists are familiar with the proximate causes of disease, that is the physiological basis of how they develop, an understanding of the general principles of evolutionary medicine would assist in gaining a full understanding and appreciation of why human diseases arise, that is, the ultimate causes. The first and core principle is that selection does not act to promote either health or longevity, but rather operates to sustain and maximize fitness. Yet clinical medicine and public health primarily focus on etiology, prevention and treatment of disease, and the promotion of health. The discordance between the focus on health and the focus on fitness is the reason why, in our experience, physicians tend to misunderstand and find it hard to keep a focus on this fundamental principle. So fitness is primarily affected by life history traits and by extrinsic and intrinsic impacts on morbidity and mortality up to reproductive age. Recent analysis of demographic data across several populations show that survival to reproductive age has a far greater effect on human fitness than age-specific fertility. This result accords with observations across contemporary hunter-gatherer populations showing that the age of puberty is markedly advanced in those populations where the rate of juvenile extrinsic mortality is high, with the average age at menarche around 13 years in some populations and over 16 years in others. The implication of the variable onset of the reproductive period is that antagonistic pleiotropy may be important in explaining the patterns of disease, in the sense that the mechanisms that have evolved to protect humans up to and during reproduction may be traded off against the adverse effects of lower regenerative or repair capacity in middle and old age. A systematic approach to evolutionary medicine, Nesi, together with Williams and later Stephen Stearns, has posed the primary question: why has selection and related processes left the human body vulnerable to disease? They identified several major explanatory pathways that, at the most integrated level, can be summarized by three factors: the inability of selection, because of its inherently slow nature, to cope with fast evolving pathogens or with novel environments, the constraints of natural selection and downsides of trade-offs, and the potential consequences of selection acting to improve fitness rather than health. Blackman and colleagues expanded this categorization to take account of the overlap between evolutionary processes and population genetics. Increased disease risk can emerge because the individual has been exposed to an environment that is beyond their evolved capacity to adapt, is entirely novel, or that poses a challenge. At its simplest level, diabetes mellitus type 2 can be envisaged as the response of the individual to a nutritional environment that gives them a metabolic load beyond their capacity to cope. While there are developmental and genetic factors that influence the adaptive metabolic capacity of an individual, ultimately, it is the exposure to high glycemic foods and a very different mix of macronutrient intakes which is thought to be the basis of the diabetes epidemic. Other infectious patterns reflect the changing environment. The historical distribution of malaria is directly linked to patterns of swamp lands and land use. Similarly, increased irrigation following the development of canals in Africa led to a considerable increase in schistosomiasis. This category combines several related evolutionary concepts that account for how the evolved human life course strategy and changed way of living have led to increased susceptibility to disease. There is necessarily some overlap with the other pathways discussed in this book, and it includes multiple possible mechanisms such as life history trade-offs and antagonistic pleiotropy. However, we find it a useful heuristic for considering a number of evolutionary explanations. In life history, there are two basic kinds of trade-offs that may arise as a result of adaptive developmental responses to environmental influences. And the first occurs when such responses are made to confer immediate advantage, such as the early metamorphosis of the tadpole of a spade-foot toad in response to pond desiccation, which promotes immediate survival but results in smaller adult size that is more susceptible to predation. The second type of trade-off arises from responses that result in an advantage that is manifest later, such as the presence of predators inducing the young of the water flea to develop defensive armor in adulthood, the trade-off being a decrease in resources for reproduction. In humans, where intrauterine growth restriction may be viewed as an immediate adaptive response of the fetus for survival, maternal ill health, or placental dysfunction, the fetus may also make anticipatory responses to more susceptible nutritional or hormonal cues to adapt its developmental trajectory to the type of environment in which, according to its prediction, it will live postnatally. There is increasing evidence for the role of developmental plasticity in influencing the susceptibility to developing disease in a particular environment. It has been shown that longevity was affected by the season of birth in the Gambia, an environment in which the weight gain of pregnant women drops from 1,500 grams per month in the harvest season to just 400 grams per month in the hungry season. Offspring born in the hungry season had the same infant and juvenile mortalities as the children born in times of plenty, but after the age of 20 they started to show an increase in mortality, such that their average life expectancy was 15 years shorter. David Barker and many others showed that size at birth, which can be taken as a proxy measure of intrauterine conditions, was associated with altered risk of metabolic and cardiovascular disease, mood disorders, and osteoporosis in later life. Elsewhere, we have extensively reviewed this area of research known as the developmental origins of health and disease, or HOPE-HAD. We review this phenomenon as a classic example of developmental plasticity operating to ensure survival, to reproduce, but resulting in antagonistic pleiotropy disadvantages in later life. It is argued that constraint of fetal growth, lower maternal nutrition, or maternal stress signal to the fetus that the postnatal world will be threatening. Many symptoms can be explained as demonstrations of evolved defense processes that have become inappropriate or excessive, and thus potentially harmful to the individual. For example, fever is an appropriate antibacterial response that activates some components of the immune system, but if excessive, can harm the individual. Similarly, a depressed mood might be the appropriate response in some situations, but inappropriate depression of mood or excessive anxiety leads to dysfunction. Fear is an appropriate response to many situations, but if the level of fear induced is excessive, or if it is inappropriately triggered, then a phobia may be manifest. The long historical exposure of humans to microorganisms such as helmintic worms is the basis of the hygiene hypothesis, which argues that since humans have begun to be reared in more hygienic circumstances, the incidence of certain diseases has risen. While the hygiene hypothesis has generally been applied to asthma, it may also apply more broadly. Crohn's disease is an inflammatory disease of the bowel which can be very debilitating. Recent evidence suggests its incidence has risen as gastrointestinal worm infection has fallen. Thus, the disease might have been caused by the defense mechanisms against gut parasites now targeting the gut wall. Indeed, there are promising clinical trials in which patients suffering from Crohn's disease are treated with either pig hookworms or their extract. Another study of patients with multiple sclerosis found that those with worm infections developed symptoms significantly more slowly than those without. The clinical trials are presently underway to determine whether treatment with worms has therapeutic value. Humans live in symbiotic relationships with a large population of bacteria, particularly in their gastrointestinal tract. Increasingly, it is recognized that this extended symbiome needs to be considered in understanding human health. Alterations in the gut flora are associated not only with acute gastroenteritis but also with chronic disease. For example, there is growing evidence that the gut microbiome plays a role in determining metabolic homeostasis and the risk of diabetes mellitus type 2 and obesity. It is not clear whether the significance of the gut microbiome arises simply from its role in pre-digestion, from the potential it has to release inflammatory cytokines, or whether it might induce epigenetic changes in the human host. A key to understanding the consequences of our relationships with the microbial world is in their fast generation times, leading to an evolution much more rapid than that of humans. This is best illustrated by antibiotic resistance. The interval between the commercial introduction of antibiotics and the appearance of resistance in human commensals and pathogens is often frighteningly short, or the order of 1 to 2 years. Broad use of antibiotics leads to rapid spread and high frequency of resistant strains, particularly in hospital and long-term care settings where rates of antibiotic use are highest. Moreover, it can be difficult to get rid of resistance once it evolves. Compensatory mutations ameliorate the cost of resistance for bacteria and can create fitness valleys that prevent reversion to drug sensitivity even after drug use is discontinued. The challenge for medicine is similar to that faced in agriculture, where insecticide use leads to insecticide resistance and herbicide use leads to herbicide resistance. Evolutionary theory has proven useful for suggesting approaches for more effectively deploying our antibiotic resources in ways that will minimize resistance evolution. For example, despite early enthusiasm, results from trials of antibiotic cycling have been somewhat disappointing. Many anatomical features of humans, such as the loss of most of their body hair, may have their origin in sexual selection. Men at all ages have a higher mortality than women, and the life history explanation for this phenomenon has been extensively discussed. Male mortality is particularly high in the early reproductive years and is associated with violence and other acting out behaviors. Such differences must be best understood in terms of mate-seeking behaviors, where the investment in competition for a mate leads to comparatively greater fitness payoffs for men. Some sexually dimorphic characteristics also impose a burden on men. Higher testosterone favors higher body mass and aggressive behavior, but is also thought to be an immunosuppressant, therefore increasing susceptibility to infectious disease. Other factors like higher somatic maintenance and faster aging in males are also thought to play a role. There is an extensive evolutionary psychology literature that aims to explain much of human behavior in terms of mate-seeking behavior and sexual competition. Unfortunately, there has been much overstatement and popularization in this domain that has harmed the overall incorporation of evolutionary thought into medicine. However, while evolutionary psychology has its limitations, the role of sexual selection in the origin of both physical and behavioral traits should not be ignored. There are many examples of founder effects and population effects affecting disease distribution. For example, blood group distribution in American Indians is dominated by the O type blood type, possibly reflecting a founder effect when humans crossed the Bering Strait. The contemporary Finnish population also descended from a founder population that underwent a tight bottleneck during migration northwards across the Gulf of Finland. It is highly homogeneous population that plays a distinct pattern of disease compared to the rest of Europe, such as being prone to multiple rare genetic diseases, but also being much less likely to develop some other diseases like cystic fibrosis. A similar situation is seen in the French Canadians, whose ancestors underwent a series of regional founder effects, leading to a characteristic geographical distribution of genetic diseases. So in sum, Erasmus Darwin famously argued that the value of what is known today as an evolutionary approach would be to unravel the theory of diseases. Charles Darwin saw hereditary disease as proof of inheritance of variation. Darwinism played an important role in biological, medical, and social sciences alike. It was also to support theories of disease that explain predisposition to a disorder as an expression of a particular pathological constitution or type, and to justify diverse social and medical initiatives. So challenges in specific features of evolutionary medicine arise from its focus on humans, because our distinct life course and the unique characteristics and the status that humans as a species have limit the range of investigations possible. An understanding of the general principles of evolutionary medicine would assist in gaining a full understanding and appreciation of why human diseases arise, that is, the ultimate causes. Applying an evolutionary perspective to clinical practice may not have an immediate impact on day-to-day therapeutic decisions, although it can lead to new clinical insights into providing an evaluative context for assessing individual clinical cases.